So my name is Shaun Wootten. I'll be furthering the discussion on Crespo et al.'s paper, colonic organoids derived from human-induced pluripotent stem cells for modeling colorectal cancer and drug testing. So here we'll start with Figure 2 in the manuscript, which further confirms the colonic identity of the derived colonic organoids through transcription profiles or markers of colonic organoids and small intestinal organoids by performing RNA-seq. So as you can see, there are several charts that are derived from the RNA-seq software, and the RNA-seq software kind of outputs these charts with with the markers and samples to further identify if it's either a colon or small intestine or the distal duodenum, depending on the figure. And these markers were derived from Sherwood et al., which Sherwood described colonic markers in a mouse study, where they described the mechanism of embryonic intestinal induction and provided methods to manipulate intestinal development from embryonic stem cells, which Crespo et al. Used in their study. So here we'll just dive into each figure one by one. So in Figure 2A, we have our color key, which we have highly expressed at the orange, so 2, and then a lower expression, if not probably not even there, at -2, which is more so the purple. So we're going to be looking at orange for the identification. So we have our colon right here, and then we have our samples right here, which is our distal, and then our small intestine organoids, and our duodenum, which you can see in here it's purple. So our colon, so it's not colon right here, but in our colonic organoids, if you look at the samples, we have high expression of our marker. So we have our orange right here. So that is more so colon. And then if we look at our small intestine, our distal, our small intestine organoids, our duodenum, we have high expression versus colon, which we get lower expression. So this further identifies whether our colonic organoids are colon or our human embryonic stem cell small intestine organoids are small intestine. And then if we go to Figure 2B, we have an unbiased hierarchical clustering, confirmed that human embryonic stem cell derived colonic organoids closely resemble human colon, whereas human embryonic stem cell derived small intestine organoid closely resemble human duodenum and distal small intestine. So we have our distal, our small intestinal organoids again, and our duodenum, and then we have our colon, and then we have our duodenum, small intestinal organoids, our distal, and then we have our colon again. And you can see right here, we have kind of this higher expression, so it's darker on the duodenum, the small intestine organoids, and the distal, which there's confirmation right here, versus the colon, we're going to have less right here, so it's not small, so it doesn't have identity of small intestine or distal or duodenum, but if you keep rising up in the chart, we're going to get a little bit darker, which further identifies the colonic organoids or the colon as colon. And then you can also use this color key histogram for the chart too. But essentially, we're looking for the darker squares as the further identification. And then if we move to Figure 2C, this is rather consistent. So we have our colon. So if we look at our colon, we have higher expression at the colon versus at our duodenum and our distal. We have higher expression at the distal duodenum versus our colon organoids, or colonic organoids, that are more representative of colon. And then we have our single intestine organoids, which is more representative of the distal duodenum rather than our colon. And then we're just using the color key again. So we have it identifies if it's orange, and then it does not identify if it's purple, essentially. So we have purple right here, so distal is not colon. But right here we have orange, so it's distal duodenum, versus colon, we have orange, so it's colon, versus purple, so it's not distal duodenum, if that makes sense. So and then we further identify with figure 2D and then figure 2E, where Crespo et al. Uses gene set enrichment analysis. And I'm not going to go be elaborate on these figures because Dr. Brockman said that due to this class not being a bioinformatics class, we're not going to fully elaborate on these figures because it's more so bioinformatics than stem cell technology, where that's what the class is. So I'm just going to quickly go through these. So we have human embryonic stem cell, small intestinal organoids, and then we have human embryonic stem cell, colonic organoids. This showed the enrichment analysis was predominantly small intestinal organoids. So that was identifies with the duodenum or the distal, where the colonic organoids were represented more in the colon by this higher enrichment score right here. And then in figure 2E, we have genes more than 50-fold upregulated in adult colon. And then we have more so adult colon, so human embryonic stem cell colonic organoids right here with the higher ES. And then we have fetal colon, which is pretty non-existent. And then we have the fetal colon right here, upregulated in fetal colon. So we have human embryonic stem cell colonic organoids right here. But we have fetal colon that rises up, so it's more fetal colon right here than colonic or adult human colonic organoids right here. So this is just further identifies colonic organoids being more colon, or small intestinal organoids being more duodenum or distal, or small intestine. So this is figure 2, and now we'll move to figure 3. So here in figure 3, Crespo et al. Derived colonic organoids with familial adenomatous polyposis, which is an inherited form of colorectal cancer caused by mutation of the adenomatous polyposis coli, or the APC gene. So Crespo et al. Created induced pluripotent stem cells from patients with FAP, or familial adenomatous polyposis, and derived the colonic organoids from the FAP induced pluripotent stem cells, or iPSCs. And then the skin fibroblasts from the two individuals created two different kinds of FAP, or FAP, which is FAP8 and then FAP9, where the FAP8 was with a 2 base pair deletion of APC, leading to a premature termination, and then FAP9 harbored a compound mutation resulting in a substitution of proline 870 with a serine. So both FAP8 and FAP9 had early termination, and we'll just get into the figures now to fully elaborate on the FAP8 and FAP9 and how Crespo et al. Derived the colonic organoids. So in figure 3a, we see FAP8 and FAP9, and Crespo et al. Confirmed the presence of the mutated alleles by genomic DNA sequencing. So you can see in figure A, we have genomic sequencing, and this confirmed the mutated alleles of FAP8 and also FAP9. And then in figure B, we have Western blotting, which showed that the wild type had higher APC expression versus the FAP8 and FAP9, which is good because the loss of APC is known to enhance proliferation. So we have enhanced proliferation at the FAP8 and FAP9 for the APC, and then the beta actin was fairly the same throughout the wild type FAP8 and FAP9. And then in figure C, we have some microscopy images of the wild type, the FAP8 and the FAP9, which just showed the further differentiated into the colonic organoids. So you can see these organoids in the pictures of figure C, figure 3C. So, and then when we move to figure 3D, we still see an enhanced expression of APC on the wild type, or just the expression of APC, and then a decreased expression of APC for FAP8 and FAP9, which still showed that there was an enhanced cell proliferation. Where then we go to figure 3E, you can see that there was enhanced staining of beta-catenin in the nucleus of the FAP colonies versus our wild type. And this was consistent with the activation of the Wnt pathway. And then you can see in figure 3E, 3F, and 3G, and also 3H, 3J, and 3L, you can see in all of these, your FAP9 has higher expression in everything that Crespo et al. was looking for, whether it was these different markers, or the Wnt pathway, or the Wnt pathway in 3G, or the mediated inhibition of RXR function. You can see this because FAP9 had higher expression due to the nonsense mutation, which resulted in shorter truncated APC protein compared to the frameshift mutation of frame 8 of FAP8. So here we see just this higher expression because it's shorted truncated APC protein of the FAP9. And then in figure 3I and figure 3K, we see just images of these organoids for this the CDX2 CCND1, and then the CDX2 Ki67, and then you can just see the confirmed colors, where green is CDX2, and then the red is CCND1, and then Ki67. So with figure 3, Crespo et al. Further confirmed the characterization of their colonic organoids, and with the derivation of Crespo et al.'s translational goal of disease modeling is to identify drugs that can reverse the pathogenic phenotype. So Crespo et al. First looks at two drugs, rapamycin and ZAV939, in Ki67 cells, and then the CCND1 cells in the CDX2 populations. And you can see when you used rapamycin or ZAV939, the percentages of the Ki67 cells in the CDX2 cells, and also the CCND1 cells in the CDX2 cells decreased with the use of these drugs within wild type, but also FAP8 and FAP9. You can see in figures B and figures D. And then with in figure E through RT-qPCR, you can see we have wild type DMSO, wild type rapamycin, wild type ZAV939, which is the first three, and then we have FAP8 in the middle that follows the same order, and then FAP9, which is the far right, which also follows the same order. So you can what was interesting about this figure was that the ZAV939 significantly downregulated the wild type through the various transcripts MYC, LEF, LGR5, GAS1, and AXIN2, while the rapamycin did not. And this can lead to potentially that ZAV939 may have some off-target effects, while the rapamycin does not. So in figure 4F and 4G, we look at geneticin, where geneticin recovers the APC protein. So you can see here at 0 micromolar per microgram per milliliter, we have 03612, 03612, and then we have 01225, 01225 for FAP9. And you can see that at the 25 microgram per milliliter, the APC has been regained. So now that we know that geneticin at 25 microgram per milliliter regains the APC, we should see a negative effect on the Wnt pathway, where we can see that in figures H through L. So you can see here, so we have the wild type and the FAP9, and you can see at this 25 microgram per milliliter, you have less, which is conducive to the APC being regained. So you can see it in K, but also you can see some Wnt effects at in figure L right here for the various transcripts again, like in figure 4E. So we have the 25 micrograms right here, the 25 micrograms right here, and the 25 micrograms right here, here, and here, and they have been significantly down regulated due to the geneticin application, which regained our ABC in figure 4G. So that primarily closes this discussion on Crespo et al.'s work, and this basically gave a modeling approach, or a organoid modeling approach, to model colonic organoids for the use of disease modeling for colorectal cancer and its potential purposes of drug screening with rapamycin and also ZAV 939 and its potential effects used in human therapeutics afterwards. And also, other limitations in Crespo et al.'s work is that the organoid model was static and was in vitro, and so it would be good to see it be used in vivo models where you have the wild type colon organoid, the FAP9 colonic organoid, and the FAP8 colonic organoid be xenographed into a amino deficient mouse, which is the NSG mouse, and then collect a colonoscopy sample from that mouse, and then have a verification of the colorectal cancer in the FAP9 mouse and the FAP8 mouse. So you could take that sample and then do a staining to verify that the FAP8 and the FAP9 mouse did indeed cause colorectal cancer versus the wild type colonic organoids that would not cause colorectal cancer. So that wraps up the limitations on Crespo et al.'s work. And if you have any questions, please ask them in the discussion board, and I will kindly answer those. Also, other avenues for this experimental proposal is to after you xenographed these colonic organoids in, you would potentially use different drugs like rapamycin and ZAV 939 to see their effects on the mouse, and you would also do a colonoscopy on those. So that wraps up my discussion. Thank you for listening.